It is in progress. All right. So I think, as you said, we were at this stage where we saved the DPF file.
Yes, sir.
Okay. All right. So after saving the file, the next step is running the grid experiment, right? So based on the parameters which you have saved, you have to run the grid experiment. So you have to go to run and run AutoGrid. All right. So uh, here important thing is you specify the program path name. It is important to specify the program path. Otherwise, the dockings will not be run. By default, only up to this thing will show in the box once you open it. So what I'll do is I'll browse because my AutoDock has been saved into by default into C drive. So you have to confirm it where it is saved in your PC. Usually it is on 86, this program 86, and the Script Research Institute, AutoDock. And then here you can see I have to run the AutoGrid experiment, right? So I'll choose the AutoGrid first, open, and it will give me the whole path of this program, correct? So this path has been given, and I have run this experiment just now. I was disconnected. I have all these files here. I will show you again. So this is the GPF file which you need to specify, that is grid parameter file. So this is the path for the file. Automatically it has taken. You can go to browse and it will guide you to the grid.com file. You can open it from here if it doesn't appear by chance. And next thing is here you have to specify a grid.glg file which will save the grid parameters, correct? So simply by putting these things here, I can launch my grid experiment. So I will launch it and you can see this kind of window will appear. It will take few minutes, maybe few seconds in some cases if you have a fast PC. So it will appear for some time, and it will, once it disappears, that means your grid experiment has been completed. For my case, it has been done. So to confirm, you will see that all these map files have been obtained here. That is a confirmation of grid file formation. And then you have to go to this grid.glg file. This is the docking log file, sorry, grid log file which we have obtained. So I can open it with the notepad or WordPad. So this is a long file. It has a lot of information. Basically, you need to go down to the bottom and it will show you the successful connection completion. That means grid experiment is successful. And then next is run the AutoDock. That means the actual experiment you are going to run. Again, you have to follow the same thing. You have to specify the path for the docking algorithm, that is browse, go to C drive, go to program 86, go to Scripps Research Institute, AutoDock, and then choose AutoDock for this time, open, right? And specify the docking parameter file this time, which will guide you directly to the default directory. And you have to put the DLG file again, same name, and I just changed the name extension here to DLG, that is docking log file. So results will be saved into this DLG file. All right, and you can simply launch it. So I have already launched it while I was disconnected, and same window will appear as was in case of that AutoGrid, but it will remain for a longer time because it is the experiment which is going on. It depends how exhaustive you have made the parameters and so on. So I will not proceed with this one, but you have to let that window go off by itself. That means it has completed the experiment. So in my case, a docking DLG file has been obtained, which should actually contain uh, all the results. It has some value here. That means it must be completed. So I will try to do the analysis by checking this DLG file. So next part is analyzing the docking results by using DLG file. So I will go and open it with Notepad, and you can screen down and see a ton of information here, right? So there are so many informations here. The most important one which we are looking for is uh, the clustering histogram. That is this one here, right? You can jump to this section, and basically it will show you this result here. So we had run 10 generations, 10 poses for our experiment, right? And you have these poses which are ranked based on the binding energy, which is in kilocalorie per mole. So the one which is having highest binding affinity, or you can say the biggest negative score, that is the conformation which is bound most tightly to the protein. So in this case, this is the second pose. That is the second model. And then that has minus 9.02 kilocalorie per mole and so on, followed by others. So basically all the poses of the same ligand has been prioritized based on the binding energy here, right? Best binding energy on the top. So this is the conformation. So if you scroll down and see, there will be X, Y, Z coordinates for all of your molecules, right? So it will show you the conformations from each cluster here. So model 2 is the top one in our case, and these are the XYZ coordinates of the model. So simply you can copy these models and open in a different visualizer, which we'll do in a minute. So second is how to open it in AutoDock tools. So simply I can go and analyze uh, go and analyze dockings, and I have to open this DLG file basically. So it contains the results, right? And then specify the uh, macromolecule and play the conformation. So basically dockings. Um, so open doc DLG file and okay. So okay. So then is analyze macromolecule. I can open the macromolecule, that is the protein, which is already here, I suppose. We can choose it. Okay, it is already here. So next is run conformations and play. So here you can see some changes happening. I will zoom it for you. So here you can see every conformation. So one by one you can see. So if you see some changes right here in the middle, I can change the style so that it is visible here. You can see this molecule which is docked in different conformation, basically 10 different conformations, right? So I prefer using Discovery Studio to analyze these binding poses, and you can do that by simply converting this .dlg file. I'll copy another file and just change the extension to PDB. PDB. So here. All right, PDB file format. So basically I will go and open my molecules, that is my protein in Discovery Studio, right? And then I can just bring this doc file in here, and now you can see this molecule very nicely, right? Which is sitting in the same binding site as specified earlier. So here you have different conformations. You can analyze one by one like that, like this, right? Okay. So these conformation are prioritized based on the score. So this is the tightly bound score. So basically, so this is one way of analyzing all the ligands, and another way of seeing each ligand one by one is saving the PDB file. What I want right now is choosing the best conformation that is from here. That is, you can copy these contents from model 2 until the end model, that is starting of the next model, end model. All right. So you can copy these contents, make a new text file, and save this file into this one, which I will name as lig pose.pdb. So here you can see this file has appeared. So I'll drag it into this window. Now you can see this has opened. All right. So in Discovery Studio, they provide you with this script, visualize close residues. So this script will take a while to run. And from here, you can visualize the residue. Basically, you can label these residues, and by amino acid name, I can increase the size. Okay, so now you can see which are the residues which are involved in binding with my pose, with my ligand, right? So for clarity, I can hide the protein. And now you can see the residues which are in close interaction. So another way, this is the 3D pose, right? You can save it as image, and then you can show the 2D diagram from here. That is from receptor ligand interactions. You can go to show 2D diagram, and it will basically give you the information on the hydrogen bonds. So here you can see, if I want to see this in 3 dimension, this hydrogen bond, I will select this pose, go to structure, monitor hydrogen bonds. And here I can see this bond, this green color. This is the conventional hydrogen bond. So this ligand is forming hydrogen bonding with tyrosine 662 of the protein. And these are other residues which are involved in different type of interactions, such as van der Waals interactions, may sea salt bridge. Then you may have some pi alkyl, pi stacked interactions, and so on. Right. So this is about the analysis of the protein by using Discovery Studio. So that will conclude this uh, workshop. So please let me know if you have questions.
Sir, if a molecule is not being bound, so it will remain lying outside, right?
Uh, how you mean, maybe if okay, there can be different reasons why it is not bound. The thing is that you are unable to define the grid around the binding site properly. Mostly that is the problem, because while you choose the grid parameters or the grid dimensions, often it can go to the ligand side. You should make sure it is on the protein side and contains all the binding site residues. And if you define the binding site, again, your ligand is not binding. That means there is some problem with the binding site conformation or the size of the grid. Either you have to increase it, the size to contain more residues to define that binding site. So most probably the thing is the ligand is not binding is with the grid settings.
Sir, actually, I was talking about a case when I don't know about the which drug is basically bound to that protein. So if I just keep on working with the different drugs, then if some drug do not bound with the protein, that means that uh, that might be a problem in my working, or it may not be binding. So if do not bind, it may lie outside, right?
Mm, no, it doesn't. Maybe there'll be some error if you are not defining the right parameters. That's it. But why it is not binding, I'm unable to. If you,
Because that drug might not be the correct one for against that protein. Is it's I'm talking about screening the different drugs whether they can bound
yeah,
protein or
if
not.
they don't bind means they are unable to bind it
So they
according to your
remain
parameters.
lying outside.
Yes, if according to your parameters, they won't bind.
And there is one more question, sir.
Yeah.
That if I have to give this figure in the paper that the binding has been done or something, so which figures am I supposed to give? Like the one you are showing right now is the final figure which can be given in a paper.
Yes, you can give both 3D and 2D because in 3D sometimes often reviewers are interested whether you have generated the right pose in 3 dimension if they are working on similar proteins, right? And 2D is representative of most of the atoms that how they are forming, which may hide in 3D sometimes. So ideally, I have seen people they can give both 3D and 2D representations.
Sir, how to save the images? Can you please tell us that the right way to save 3D and 2D images that can be given in paper?
Yeah, so uh, I hope you are still seeing my screen.
Yes, sir.
All right. So using Discovery Studio, you can do it. For example, if I am nicely setting up my pose here, I'd like to change the color of my ligand. Suppose carbon only apply to green. So you'll see nice and green color residue. It is connected, so you can see changes on the right hand side also. So first thing is I want to save this 3D pose. So file, save as, you'll have information down there. I want to save it as image. So by default, it is in next gen helper, the same directory. So save it. If you want to save a larger image, you can change the width. Maybe around 2000 is good enough. So automatically height will be adjusted. Okay. All right. And similarly, you have to click on the second window. You have to activate the second window. Currently, I was here. So you have to click on this window, 2D, so that it is selected and go again and save it as such. Okay. So you have options. I want it in image file. So I will choose 2D. Okay. I think I'll rename the previous file. Sorry about that as 3D. And this I will put it as to save. Again, it will ask you maybe 1500 this time. You can check the quality. Let's see. Okay, so 2D is saved here. I'll open it. So you can see nice and clean picture. So the next one is the 3D pose like this. So there are a lot of things you can devote some time to learn Discovery Studio. So maybe changing, such as changing the background to white. Okay. And yeah, moving these around, like you can move control and uh, left mouse button, you can do that, right? So that's all for this session.
Thank you so
Any other,
much, sir. Thank you.
any other question, you can email me, right? I'm available.